This is the third and last of the recordings regarding Chapter 18, which was the subject of blood and the circulatory system. I hope to finish up Chapter 18 here, so 3 lectures. This is all for, as it says on the screen, Lecture Exam 3. And as you will, I hope, have soon learned, we're going to go back as best we can to live lectures and meeting in the classroom. So this might be, at least it's my hope, that this might be the last of the recorded lectures for Human Anat Phys 2 in this spring of 2022. We're continuing our look at blood and some of the details of blood. And today, I want to talk about blood types and then end by talking about blood clotting, how blood clots, and then all kinds of other disease or health issues related to improper blood clotting. But let's begin by talking about different kinds of blood types. So chances are you know your blood type. If you don't know your blood type, that would be a good thing to do. And you might wonder, why would that be a good thing to do? Well, it's good that when you're in a medical situation, we know what your blood type is so that if you need blood, we match it appropriately. And we're going to learn what that looks like. So I have blood type A negative, which is kind of a rare blood type. And maybe you know what yours is. Or maybe at the end of this lecture, you're going to be curious to see if you can find out. It might be on a medical chart of somewhere in a doctor's office that you have, or any other kind of medical documents that you have. At hospitals, my understanding is that they check your blood type before they go in and do a transfusion. But sometimes on a personal driver's license or another place on a personal identification card, you might indicate your own blood type. So let's talk about the different kinds of blood types. Let's talk about what makes the blood types that they are, and the actual details there so that we can understand compatibility. And we'll talk about why it would be a problem if you mixed certain blood types. Okay. So blood typing and transfusion is all about specific interactions between proteins and red blood cells. Okay, it was discovered, the major blood types were discovered about 120 years ago, 122 if it was 1900 even. And those blood types you may know are A, B, and O, which is odd because it's really 0, as we'll learn. It's A, B, and 0, but everybody calls it O. Probably because the other two designations are letters. We'll see why 0 actually makes more sense than O. Dr. Landsteiner earned the Nobel Prize for blood typing in 1930, and of course this would have been really, really important in World War I when we didn't know any better about mixing blood, except that many times it caused problems, and certainly it would have been helpful in other future wars that followed, where people need all kinds of transfusions. And of course, in just plain old personal medicine, in our lives, any one of us can be in an accident in the next couple hours and need a blood transfusion. Now, these kind of interactions that we're talking about are based upon antigens and antibodies. And remember that we're talking about antigens and antibodies regarding, as it says above, plasma proteins and erythrocytes. This just in. So I am recording this on Friday, February 18th, and just today it was reported in the news, this surprising, and I think quite surprising, announcement. If you know about blood typing, you'll know that that would be an issue then when you donate and you have a donated organ, so when there's a transplant. And one of the problems is that you have to match up the compatibility. That's one of the factors that we look at when we're trying to get organs transplanted. And as you can see, people have experimented here using different enzymes, and they were able to switch a lung from type A to type O. And type O is the universal recipient, as we're just about to learn about. So you can freeze this and read more about it, but it's really, really quite exciting. And we'll have to see how this plays out. But it seems like we are now understanding the mechanism to do this, and that this may be another way that we can increase the number of organs available for transplant. Excuse me. Antigens and antibodies. Let's make sure everybody understands what antigens and antibodies are. Antigens, as it says, are surface molecules on membranes that elicit or cause an immune response. It doesn't have to be molecules on the surface of cells. Some people are allergic to other things, including, as I understand, nickel, the metal, nickel. So lots of things can be an antigen and elicit an immune response. Generally, allergies are such a thing, whatever it is you're allergic to. So it could be latex on a glove, right? That could be the antigen. Antigens, if they're molecules on the surface of a cell membrane, are then genetically unique to the individual, as it says right here. And our immune system is monitoring self from non-self. So we're always trying to figure out whether or not something is foreign, something is not of us. And of course, that could be lots of germ particles of sorts, infectious agents, or pathogens. So these antigens are generating an immune response. Here you can see a specific one regarding blood typing, and those are called agglutinogens. So agglutination is sticking together, and gens is to create. So creating, sticking. And these agglutinogens are antigens on red blood cells that then are going to be the important factors that we're going to consider for blood typing. Now, antibodies are produced here, you can see, by plasma cells within your immune system, and antibodies are very specific for certain kinds of antigens. So when we were trying to elicit an immune response against COVID virus, which is SARS-CoV-2, we were picking certain antigens, certain molecules or parts of molecules on the viral particle to try to get our immune system to recognize SARS-CoV-2, the virus. Okay. So antibodies are being produced by our body, and they're very, very specific to the antigens. So you can see antibodies are helping to identify antigens and mark them for destruction. When antigens bind to antibodies, these form a complex. Clearly, it's not a big surprise, antigen-antibody complexes. So we could have antibodies attacking the surface of foreign red blood cells. That's the setup here. To this point, you have a mental imagery of someone's foreign red blood cells that clearly have markers that say they aren't you, and your own immune system then attacks those foreign red blood cells. Agglutinins then are these antibodies that are going to be cross-reacting with foreign red blood cells. This agglutination, I said already, is the binding together of some kind of antibody that's binding to some kind of antigen. And what that would do is cause a clumping of red blood cells. So imagine an antibody joins a single antibody joins to two red blood cells. That would cause those to stick together. And this clumping of red blood cells is the agglutination that we're talking about. So now let's talk about what are the factors. And here we go, looking at the ABO or AB0 various groups. Our immune systems might produce antibodies to the antigens, the antigenic molecules A and B. Okay? And your antibodies that are going to react to blood type A or blood type B, as you can see, appear pretty much in the first year of life and grow till about the first decade of your life. These antibodies are found in the plasma, and of course these antibodies are binding to antigens on the surface of red blood cells. In general, you don't form antibodies against your own antigens. It's part of the maturation of the immune system, in which the immune system is learning its chemical reactions. It's not really a thoughtful process, but learning to identify self and non-self. Now, you should have remembered that we have talked about autoimmune diseases previously, Talked about them in Human Anat Phys 1. Autoimmune diseases are when our own immune system then, for some reason, starts attacking ourselves. It could be that there's some cross reactions where an antibody response against one kind of foreign agent cross reacts with something in your body. And other experts on the immune system could probably go on more about what are the known causes of autoimmunity. And it must be a growing list. But this idea that we do not attack ourselves, how we learn to do that is something you would learn in immunology with Dr. Welch. And also the failure of that then is the generation of these autoimmune diseases, which can be really quite serious. Now, this is table 18.5 in your textbook, and I'm flipping through the pages right now. See if I can make sure I can find where 18.5 is located for you. 18.5 is on page 683. And this is a really, really important table. So really take a look. If you're type O, what that means is you do not have any of the A or B antigens. That's why it's 0. Type A blood is known for its antigen on red blood cells, and that's the A molecule on the surface of red blood cells. Likewise, B is a different antigen on the surface of red blood cells, and someone with AB blood type has both A and B antigens on the surface of their red blood cells. Now, if you have an A antigen, you are not going to have antibodies to A. That doesn't make any sense. If you have the A antigen, you don't have antibodies to yourself, but you do have antibodies to B. Likewise, if you have B antigens, you don't have antibodies to yourself, but you do have anti-A. If you have both A and B antigens on your red blood cells, then you are not producing any antibodies related to A and B. And now come back to type O blood. Because neither antigen is present, type O blood then doesn't have any antigens for these guys to attack, but instead type O blood does have anti-A and anti-B antibodies. Okay? So, who may successfully receive type O blood? And the answer is you can only receive type O. You can always receive your own blood type. Type O blood, because it doesn't have any of the antigens regarding these factors, A and B, type O blood then can be donated to just about anybody else because there's nothing to attack. Type O is missing the A and B, and so if you have an antibody to B, well, type O doesn't have any antigen. Likewise over here and here. So it's always O and whatever it is you are. That's what you can receive. How about who can you donate to? So O can donate to everybody. A can donate to A, and A can also donate to people with AB blood type. If you have AB blood type, you have the antigens but no antibodies. So an AB blood type can receive from everybody. So O is considered the universal donor. AB is the universal recipient. So that's pretty cool. So type AB blood, you can get, because you don't have any of the antibodies, the AB blood is not going to be reacting to anybody else's blood. O will be reacting to everybody else's blood, so they may only receive O, but because they don't have the antigens, nobody's going to be reacting to O. I've found that over the years, and I've been doing this a while, so this is probably at least the 35th time that I've taught this class, I've found that this kind of stuff, when you really think it through and you get it right in your head, you kind of go, okay, And then somehow quickly, at least I find myself doing this sometimes, I get just a little confused and I have to go back and check this. So all I'll tell you is this is tricky in that I think that there are times when it makes perfectly good sense, but then you come back to it in a half an hour and you kind of go, oh, wait a minute, okay. And that's why I'm telling you that this particular chart, telling you about who has what antigens, who has what antibodies, and if you recognize you don't want to donate a foreign antigen to someone else, right? So here, if you're anti-B, you certainly wouldn't want B blood coming into the body because they have the antibodies to B, so B would not be a good donor here, right? And then you have to figure out the usual things about O being the universal donor and AB the universal recipient. Now, the frequency of these blood types varies tremendously in the U.S. population. I'm guessing these stats are from different people of different backgrounds who live in the United States. It's really quite interesting. So if you look type O blood, very, very prominent in Hispanics. Native Americans, type O, very, very prominent, and usually so. Then notice the counter very, very low numbers over here. So a Native American of AB blood type would be really, really rare. Okay. Type A blood that I am, well, I'm white or identified that way, Caucasian, and about 40% of white or Caucasian, probably European ancestry, are type A. So it's very, very common. It's not all that rare. Here, type O, seen most commonly here in white people. So with people who are black, identified as black, here the most common is type O. Type O, as you can see, is pretty much the most common, with the exception of Japanese, in which type A is more common than type O. So that's interesting. So pretty similar for white, and then we start noticing bigger differences, a really big difference for Hispanic on down the line. Again, big bold. The type of blood you are is the type of antigen you are. I would begin with that. Now, in my experience, if you keep track of that, type A has A antigens. Then you can derive the rest. Okay, A is going to have A antigens, so that they will only have B antibodies. Okay, that makes sense. And then you keep thinking about that. Okay, if you got B antibodies, then clearly you can't be accepting B blood or AB blood. That doesn't make any sense. But with having anti-B, so antibodies to B, but type O doesn't have any B antibodies, so they could be a donor to you. That's why A can receive A and A can receive O. I'd look at this, I'd study it, I'd come back to it often to make sure it makes sense to you, okay? So these are the things we've already covered and addressed in the table above. Keeping track of the antigens informs then the type of antibodies. So the universal donor, type O. The donor's plasma for type O could have antibodies, anti-A and anti-B. That's usually diluted and not a factor in the donation, but it could be that they're going to pack the cells, which means they're going to minimize the plasma and minimize the antibodies which are in the plasma, right? That's why that makes sense. Universal recipient, AB, unfortunately it's the rarest. Those people can receive just about anybody's blood regarding the A and B antigens, because we're about to talk about something else. So what blood type can be given to everyone else? It should probably say be given to everyone else. And the answer is O, right? That's the universal donor. What type can receive from everybody else? That's the universal recipient, and that's AB. So now, here's the question. So what's going to happen? Well, if you give the wrong blood type to the wrong person, you're going to get agglutination. You're going to have antibodies binding together red blood cells. Each antibody can attach to several foreign antigens on different red blood cells at the same time. This causes the agglutination of red blood cells. As you can see, all sorts of bad things are going to happen. You're going to block blood vessels. You're going to rip apart red blood cells. They're going to release hemoglobin into the blood, and we already talked about that as a risk to the kidneys. Okay, so hemoglobin blocks kidney tubules and could cause renal failure. Here's a figure. This is 18. Actually, this is 18. I think it is 18. Yeah, it's 1815 in your current 8th edition of your textbook. And so this is just showing a type B person receiving type A blood. And so they're interacting. Red blood cells are agglutinizing, so they're joining together and they're clogging things up. Okay, so that's it for type A, B, and O blood types. But in addition, there's another factor. And the agglutinogens, it's called agglutinogens, RH, the rhesus factor, were discovered in 1940. So RH factor is how you probably know it. And blood can either be Rh positive if you have the antigen, or Rh negative if you don't have it. So if you have it, you do not have antibodies to Rh. If you don't have the Rh antigen, you have antibodies. Now, I told you I was A negative. So if one of you had volunteered your blood type, we'd do it with you, but I'm kind of by myself here with Lucy, and Lucy's not talking because she's asleep. So I have type A blood. What type of antibodies do I have regarding A and B? And the answer is I have antibodies to B. I'm Rh negative. So do I have antibodies to the Rh factor? Yes. So I have antibodies to B and to the Rh factor. Now, I don't really have a lot of them. It's not ramped up. My immune system's not producing a lot of those, but it could if in the presence of these antigens. Rh factors vary just as well. I don't think it was in that last table. And then just as well here, why Rh? Where'd the Rh come from? And the answer is the Rh was discovered in rhesus monkeys. So this is a rhesus monkey. And this is a different kind of rhesus, not rhesus monkey here, but over here, this kind of rhesus monkey would be this. So don't be confused. The thing on the right is candy, related to candy. The thing on the left is related to blood type. And let me just tell you, this is why we have to meet in person. Because if this was in person, I would be watching you guys wipe tears from your eyes because you're laughing so hard. Not. All right, moving on. Well, what's the deal with Rh factor in pregnancy? Well, if a woman who's going to get pregnant is Rh negative, does she have the potential to produce antibodies to the Rh factor? Yes. So, she normally doesn't have these antibodies. That's what we're talking about. That's the anti-D-agglutinins. That's the Rh antibodies. They're normally not present. They form, as I was just telling you, in large number, significant numbers. That is, to the level that it matters for someone's health. They form when exposed to the Rh factor. Okay? So if an Rh negative woman has her first baby, who's or has a baby, eventually has a baby that's Rh positive, or, of course, if she gets a transfusion of Rh positive blood, the first transfusion or the pregnancy probably isn't going to be a big issue because then she ramps up the Rh agglutinins. The problem is if a mother formed the antibodies and is pregnant with a second Rh positive child. So in the first pregnancy, when she's exposed to some of the child's blood, which might be during birth, for example, then she would ramp up these antibodies, but probably not of significance to the first pregnancy. It's that she is negative and now is pregnant with a second Rh positive child. That's when her immune system has the potential to be attacking that second child. So this is HDN, hemolytic disease of the newborn. As you can see, the antibodies or agglutinins can cross the placenta. And so how do we do this? Well, what we often do is when a woman is Rh negative and she has an Rh positive baby, what we can do is give her something in her bloodstream that binds to the antigen, to the Rh factor, so that she does not start forming the antibodies. So we try to prevent this learning experience, if you think about it that way. We try to prevent the first stimulation of the Rh antibodies by blocking the antigen. That's what we're trying to do. So here's a figure in your textbook. This is 1816 in your textbook, and they're trying to explain. So here's a Rh positive fetus, mom's Rh negative, and she's already given birth, so first pregnancy goes fine. And between pregnancies, what's she doing? She's producing a lot of antibodies to the Rh factor. And now the second time she gets pregnant, her immune system is going to produce antibodies to her fetus that'll cross the placental barrier and potentially kill the second fetus, the second child. This would be the second Rh positive child. You can go back and take a look at that in the textbook. Clarify it if you need to. Moving on now, beyond erythrocytes. So we're done talking about erythrocytes. And I want to say just a little bit about leukocytes. So if you're a member of the plasma membrane bound structures in blood, we have erythrocytes, we have leukocytes, and then there was going to be a third membrane bound structure in blood. It's not an erythrocyte, and it's not a leukocyte. What was it? And the answer is platelets. We're done with erythrocytes, and I want to tell you about leukocytes just a little bit, which is it's very diverse in your textbook, and I'm not going to say much because the textbook does a great presentation. Leukocytes, the white blood cells, very important in their roles in the immune system and fighting infections and removing dead and damaged disease cells. So important. And immunology is such an important topic now, including the immune system attacking cancer cells. Okay. These leukocytes had a very clear and distinct nucleus, and that's not the case for a human mature red blood cell. Leukocytes don't spend a lot of time in the bloodstream. They enter the bloodstream and then generally migrate out into connective tissues. They're retaining all of their cellular function, and they're able to make a lot of proteins. These accumulate sometimes into what we call granules, and all white blood cells have lysosomes, of course, that allow them to engage in phagocytosis. Granulocytes, so eosinophils and basophils, for example, and neutrophils, have these, and we say granules, but you remember we really mean vesicles that contain enzymes, other chemicals that are related to their role in disabling pathogens and other kinds of disease cells. But what I want to say is this. That's the basics. Your textbook, the 8th edition, has 6 really, really good pages about leukocytes. Of course, it's assigned along with the rest of the chapter. You can very thoroughly compare the structures and functions of these different kinds of leukocytes, so you have a basic understanding. You will need that kind of basic understanding if you go on into immunology, and students that are going on into medical technology or going on into nursing will absolutely need to understand the role of the different kinds of leukocytes. But this is all so straightforward on these pages, I don't see a point in me talking about it with you. So the pages describe the leukocyte life cycle or the life history, notes disorders, and as I said, this is all very clear, very well organized, and does not require our time to discuss. So I'm not going to say that much more about leukocytes. and we're going to move on and take a look at platelets. So I'm talking about blood clotting now, and this is the end of us talking about the circulatory system blood chapter 18, by talking about clotting. So if you want to take a break while you're listening to these, this would be a good point to pause, because we're shifting here to talk about blood clotting, and that is the end of this lecture. So we said that platelets were the circulating factor that was involved in clotting. And I also mentioned to you that platelets are actually pieces of megakaryocytes. And here's actually the basic relationship. You have a megakaryocyte that's outside of a blood vessel, and it has these extensions called protoplatelets, these extensions that extend here within the bloodstream, and they kind of get knocked apart, and pieces break off here. And so here we've got platelets breaking off of some of these proplatelets. And that's the actual platelets that are circulating, okay? And here we saw these platelets indicated in this general art about the different types of blood cells and the particles that are in blood. The platelets and their involvement in clotting is limited because if we have a very, very large bleed, a very large blood vessel, these clotting mechanisms will be insufficient. And so a very large blood vessel, usually an artery because it's higher pressure, a very large artery that's damaged, a person could lose so much blood that clotting isn't going to happen, and eventually they're going to die. And if this is bleeding into the abdominal cavity, so into the coelom, there may be coelomic distension, so the belly looks full and it's getting very tight. And that's because the coelom is literally filling up with blood. And that, again, is a life-threatening problem. So in medical care of some sort, if you're an emergency person, or whether or not you're working in an emergency ward, or you're in a situation where people could be really hurt, internal bleeding like this is a real issue. Because if it's a big blood vessel, it's not going to clot on its own. So here are a few basics about platelets. Second most abundant formed element in blood. And so what do you think is the first? What is the first or most common formed element, which means it has membranes around it, in blood? And by far it's red blood cells, right? Erythrocytes. We said that these break off the megakaryocytes. We already just talked about their breakdown, or their breaking apart of proplatelets, an extension of the megakaryocyte. Platelets tend to be 2 to 4 microns, and I'm curious if anybody remembered roughly the size of a red blood cell. And this isn't, in my mind, quite drawn to scale, because if they're 2 to 4 microns in diameter, the diameter we said for red blood cell would be somewhere between 7 and 8, and the book mentioned 7.5, but I said it depends on the treatment of the red blood cells when you're making the sample. So I use 8. And so if it could be that these are only a quarter of the diameter, but if it's 4 microns, well, that's half the diameter of a red blood cell. And I'm not seeing platelets here that are half the diameter of a red blood cell. So I think the scale's off just a little bit. Not a big deal. We find megakaryocytes in red bone marrow, and you can see then that they're going to have these extensions into blood vessels that then are blood vessels that are invading or within the bone marrow. Talked already about a little bit about how the so isn't that interesting? I think I'm seeing both protoplatelets and proplatelets. Huh. Well, now that I'm lecturing about this, guys, I got to tell you, I don't know if there's a difference between a protoplatelet and a proplatelet. Huh. I'm just looking really quick in the textbook to see if I'm seeing any kind of distinction. Yeah, I could have written it down. It may just be proplatelet. Anyway, until you hear any differently, I'm thinking that protoplatelets and proplatelets are the same thing. Platelets last, as you can see, for about 5 to 6 days. Why? Well, because they don't have a nucleus and they don't have the machinery to replace themselves, so things are going to break down. Where do we find platelets? Well, a quarter to 40% are stored in the spleen. So that would mean somewhere between 75 to 60%, or 60 to 75%, are circulating in the blood. That's right, the rest are in the bloodstream. But as we've been talking about the circ system, we've mentioned the spleen a few times, including some of the breakdown of hemoglobin. And if you've heard of someone who has a damaged spleen, and it's often in some kind of accident, often where the chest is heaved forward. So for example, you're riding a bicycle or a motorcycle or a four-wheeler, and the handlebars crush into your chest as well as just below your ribs, so below your diaphragm. And you could rupture your spleen, and somebody's spleen might be removed. And so, can you live without a spleen? Yeah. What do you lose? What are the functions you lose? Well, that would be a good test question, wouldn't it? And here you would lose reserves of platelets. So we keep some platelets in reserve here in the spleen so that if we have a major clotting event, this extra store of platelets could be released. But if you don't have a spleen, well, that capacity then doesn't exist. Platelets have within them vesicles, they're calling them granules, and there's lots of secretions that are in them, special different kinds of molecules that are important in the role of clotting, and we're about to see that in a couple slides. So I'll show you that list. That list also exists on page 694 in your textbook. You know what? I'm actually going to stop for a second and see a proplatelet. Yeah, hold on. Yeah, I think I made up the protoplatelet. I think that was just a mistake on my part. So I just looked in the index of your textbook, and I'm not finding the word protoplatelet. So I think they're all just proplatelets, just like it says here. Okay. What can proplatelets do, or in this case, platelets do? Proplatelets are breaking down into platelets. What can platelets do? Well, they actually can creep along a little bit. They can actually react to their environment and engage in amoeboid movement, and that's where the cytoplasm kind of oozes out and creeps along a little bit along probably a damaged area. Also, platelets can phagocytize bacteria, so they can actually be involved in getting rid of bacteria. Here's one of these really neat photos where they highlight a megakaryocyte. You can see that they do, in fact, have a giant nucleus. And the textbook talks a little bit about DNA and chromosomes multiplying, but not dividing the cell, so that it just gets this really big nucleus. And didn't really talk about the functional significance, or I didn't see that, but I would guess that that's an increased capacity for transcription, so that more of the molecules that are necessary to be produced that are going to help platelets, the capacity of this cell is increased. So there can be more DNA available for transcription, more mRNA for translation, and that means more proteins. I would guess that. Not I would guess that, I just did guess that. All right, so I promised you on page 294, here's the list that's right there. There's no need to copy all this down. It's right there in your textbook, but let's talk about it. So platelets are going to do a lot of things. They're going to release some substances. By the way, when they're activated, so we're going to talk about this. You wouldn't want platelets doing clotting functions if you don't have the need to clot, right? So I'm just about to talk about that in a few minutes, where inappropriate clotting mechanisms can actually lead to serious problems in your body. It's a balance between getting ready to clot and engaging in clotting when it's needed, and not pulling the trigger too early, not causing clotting when it's not needed. So once activated, It's going to promote vasoconstriction, so some of that smooth muscle on blood vessels is going to constrict down. It's going to help pull together platelets, stick them together, as you can see, to form a small plug. Other coagulant factors that are going to, of course, promote clotting. And then, interestingly, there's also some clot dissolving enzymes that are there, so that it can kind of, like a sculpture, you're adding in one place and subtracting in others, and allows it to probably be a better fit for the damage. We're attracting neutrophils and monocytes. That's great. There's some destruction of bacteria and growth factors that are promoting already the repair. So it's really doing a lot of things. It's stopping the problem and promoting healing. So this is the hemostasis. This is the control of bleeding and making sure it doesn't go off in the wrong way. And there are three main mechanisms right here that we're going to talk about. So we're going to talk about when you have some damage to a blood vessel, the initial reaction called a vascular spasm, and then platelets forming a plug, and then the actual bigger plug formation going on later on. So platelets plugging things up and then forming a true clot. Platelets are involved in all. So let's take a look at this. Here in figure 1821, and that is on page 695 of your 8th edition of your textbook. Here you're seeing those three events that we just listed: the vascular spasm, the platelets forming a plug, and then the actual coagulations going on. So notice this is a network of fibers, and we'll talk about how that forms. And this is just the platelets to begin with. So that's clearly a separate event over here. So let's go through these stages. The vascular spasm is then the constriction of these blood vessels. And we said this is only really going to help if it's a very small blood vessel. If it's a great big blood vessel, this isn't going to be sufficient. And so the very first thing when a blood vessel like on your skin is broken, so you've cut yourself, is it's going to decrease the blood flow. And if you think about it, if you've cut yourself and it's not really bleeding much, could you then squeeze your finger and get it to bleed more? So one of the little jokes and things that we've talked about before is little kids sometimes get hurt and it's just barely bleeding, and they've learned to squeeze their finger to make it bleed more so they can get more sympathy from mom and dad or whoever is around. But that, if you think about it, you're cut, it's not bleeding much, but now you're squeezing it and you get more blood to come out. That's probably due to the fact that there's already some constriction here. And yes, it could bleed more if you forced it, but already we get that immediate constriction so that we reduce blood loss. Interestingly here, what triggers vascular spasm? And right away, it's some pain receptors that actually are innervating blood vessels. And so when it hurts, the blood vessels are constricting. Smooth muscles get injured. Platelets in turn, so smooth muscles that are injured themselves are going to constrict. And here now the platelets releasing serotonin. And serotonin, so look at your word roots. Vaso means blood vessel. Constrictor is constrict. So you would figure that out. So platelets could be releasing that. We could have lots of other things coming along. So constrictions of blood vessels are then happening. And what I said was pain receptors would right away, I mean that's immediate, cause some constriction. Smooth muscle being injured is going to take a longer period of time for constriction. But these two start things in action. Platelets are also contributing, but platelets are going to do a lot more. So let's take a look. So immediately you get some smooth muscle constriction, and then you have the chance for platelets to start doing their thing. So step 2, platelet plug formation. So for starters, why aren't platelets always sticking together? Is it that they're like, chewing gum or some kind of glue or something? Is it that they are always sticky? Well, they always have the capacity to stick, but what they stick to is usually exposed, so they got to listen, what they stick to is usually exposed when there's damage. So a normal healthy person should not have a lot of material exposed to platelets that causes platelets to stick. Intact vessels, the lining of intact blood vessels, have a smooth endothelium coated with this molecule, which is a platelet repellent. So platelets are not normally sticking to the inside of blood vessels. That's what you need to understand. But when a blood vessel gets broken, there's collagen that gets exposed. And that now starts a lot of this plug formation. So little extensions of platelets stick to the damaged blood vessels, and they also stick to other platelets. They all start attracting and pulling together in like a drawstring. And so they literally can shrink together and really form a compact clot or plug. And you can see it usually takes about 30 minutes. So I don't know if you have a normal good clotting going on in your body, if you've thought about the fact that you have something bleeding. Could be on your face, could be a zit, whatever is bleeding, and how long is the darn thing going to bleed? And this is a process that takes, depending on the size of the injury, maybe up to 30 minutes. So I shave with a blade. I'm not sure why. I guess because it's closer than an electric shaver. And for whatever reason, I've been cutting myself more often. And right away it starts bleeding. It's not a great big opening. In fact, it's a little bitty thing, but it bleeds because it's on our face and we have blood vessels very close to the surface. And what I'm going to tell you is what you have to do. You do not keep wiping it because you're wiping away a lot of these plugs. You might dab it a little bit. And some people might take a little piece of Kleenex or toilet paper and actually put it on the bleeding part and let it just stay there for about 30 minutes to an hour, in which then you might want to wet it and gently take it off so you don't break the plug or the clot that's forming. So right here, this initial platelet plug is delicate, so you don't wipe a bleeding injury, but you could blot it a little bit if you wanted to remove some of the excess fluid. Okay. As the platelets form the plugs, they degranulate, which means they release by exocytosis a lot of substances that were in these vesicles. Okay, a lot of substances that are in the vesicles. And what are in the vesicles? Well, we mentioned serotonin earlier as a vasoconstrictor. So that's one of those initial roles that we were talking about on trying to cause blood vessel walls to constrict up, to reduce blood flow to an area. They also release ADP, which is adenosine diphosphate. You know ADP as the molecule that binds with phosphate to make ATP. That's my understanding. I hope I don't have this wrong. And this attracts and causes platelets to release more of their substances. So it's a positive feedback. The result encourages the process. And oh my gosh, look at all the other kinds of things that come spilling out of these vesicles of platelets. So I mentioned it's positive feedback. The platelets degranulating causes more platelets to degranulate. And now they're releasing lots of these substances in that immediate area of the break in a small blood vessel. Coagulation, step 3. So the plug has formed, and now we're looking at the chemical reactions that form a much longer lasting clot or scab, as you might know it. Last and most effective defense against bleeding. So before we go on, why would we not want clotting to occur in the absence of bleeding? Well, because in fact it's potentially going to clog up blood vessels and clog up flow. And I'll talk in detail about that in just a few minutes. This whole step 3, coagulation, is unbelievably complex. In fact, I can imagine in molecular biology or biochemistry that a person could have more than one lecture just on step 3, this coagulation process. Lots of chemical reactions. I'm going to show you just some of the most fundamental basics of this and tell you that you are not responsible for these high-level details beyond what I'm just about to tell you here. So this is good news for you. I'm not going to be expecting you to know the kind of extensive details that you will see on page 697. So if you look at figure 1822, which I'll show you in a second, those kind of extensive details are not something you'll need to know. Coagulation involves, so these are some of the basics you do need to know. Coagulation involves the conversion of fibrinogen into these threads, fibrin threads, that are actually, so if you go back to that figure that showed the difference between the initial clustering of platelets and then the actual formation of a clot, I pointed out that there were threads or a network there, and that's these fibrin threads that form. And they're sticky, and they're forming the framework, as it says, of the clot. So there's this fibrin network there that's helping blood cells and platelets to all stick together and clot up whatever the damage was. And my analogy is it's like insects getting caught in a spider web. Okay? So here's one of the figures, and this is part of the figure on page 697, figure 1822. So what we get, as you can see then, and what this is the fibrin, so the fibers, blood cells, and platelets all clustered together. And in theory, that's going to seal up whatever the damage is to this relatively small blood vessel. So what we have is chemicals that are released by the liver that are already in the plasma. So one of the many things in plasma, clotting factors sometimes is generally what we say. So I might have listed that before as a component of plasma, and those are clotting factors. And we said before that serum is plasma with these clotting factors removed. Okay. Well, the clotting factors here are in an inactive form, and once activated, this is really the point about this being very complicated, it results in a complex chemical series, a series of chemical reactions that we call a reaction cascade. And that's where the product of one reaction triggers the next. And just like going down a waterfall, each step hits and impacts the next step. So within clotting, two pathways: extrinsic, so the tissue itself being damaged initiates some of these reactions. And then intrinsic is as blood is itself starting to now clot and coagulate, it triggers these other internal reactions. So there's an external trigger, and that is the actual damage, the exposure probably of collagen and all the reactions of platelets. That's the extrinsic. And then once it gets going, it continues and encourages itself. That's the intrinsic. Okay. And this now is the full figure of 1822, which is showing you way more than you're going to need to know. It's beyond the class, and it's very important, and my question to you is why? Why is it important for people in medicine and research to understand all of these steps? Well, it's a long answer. If you're trying to come up with a drug that's going to interrupt the clotting process, then these are all the potential steps where you can interfere. Here's the extrinsic and intrinsic that we just talked about. This is, you got damage, and so the external damage itself is causing reactions. And then once we get a clotting reaction going, it's self-encouraging. It's a positive feedback. If you look at any of these steps, could you come up with a drug that would interfere so that if we have too much clotting going on, we can then decrease the likelihood of clotting. And some of you may know that aspirin actually is one of those factors that can decrease clotting. And there are many others. We'll talk about them a little bit. So if a person is at a higher risk of having clots formed when we don't want them to, and that can happen from people being very sedentary, sitting around a lot, being very inactive, and of course that could happen by a person who's hospitalized or immobilized because of their own health situation. We might put them on some kind of clot reducing drug or medicine. Another way that this really matters is, what if somebody has a disease and in some way some factor's not being produced? And you may know about hemophiliacs. And so hemophiliacs could have something in this pathway that is in fact not working, and in them, even when they do have damage, they don't trigger this pathway properly. But all of those fine details are really beyond the class, so that's good news for you. I want you to know the basics that we just talked about before, the understanding of an extrinsic mechanism versus an intrinsic, and that involves pathways, that ultimately we're forming this kind of a big clot. But you don't have to know all these enzymes and all these factors. Okay. So, we got a clot going on. Platelet-derived growth factor stimulates fibroblasts and smooth muscles to start the repair and repair the damage. And then later on, we have then the dissolution or the breakdown of a clot. Okay. So what happens to blood clots? Well, as they exist, they're actually stimulating repair, so that's good. And then eventually they're helping to dissolve themselves. There's also, if it's on the surface of your body, there's also just some wear and tear. So washing your hands, getting it wet, and some friction will reduce the size of clots. But generally, it's a good idea to leave them alone. And some of you undoubtedly have had clots. I have. And either I picked them off because it was itchy, or I knocked them off because it was in a place that I was getting beat up a lot, like, your knuckles or your hands. And then it bleeds again. And then, darn it, we got to start the whole process over. These are the details of breaking down a clot. And this is a detail that is, again, beyond, in my mind, this course. So this is figure 1824 on page 699. And you need to understand that there are mechanisms that break down the clot, but you don't have to know all of these steps. Thank goodness. So, what keeps clots from forming when it's not adaptive? Well, platelets generally don't adhere, we said already, to the lining of the endothelium. So that's good. And they generally don't stick to each other. Further, thrombin, which is going to be promoting clotting, is diluted. So wherever thrombin's being produced, it will wash away and not be in high concentration, as long as you got a good circulatory system. But interestingly, people who have a very, very slowed heart, perhaps because they're in shock or other kinds of problems that are going on, run the risk that this thrombin will actually build up to a level to start triggering some clotting. We have natural anticoagulants, as it says here, and you can see it's heparin, and there are heparin drugs that people can take. And then there's this antithrombin that you can see comes from the liver that actually helps to be in balance and keep thrombin from overreacting too early. What happens when things go wrong? Well, this could be really, really bad. In the case of hemophiliacs, and again, there are, although you don't have to know them, you should know there are many different kinds of factors that a person could have a problem with. So there's different types of hemophilia and affecting different kinds of factors. So it's not one disease, okay, all caused by the same thing. It's many different factors. In some cases, it's a sex-linked recessive. So in the case of sex-linked, if women have 2 copies of a gene and men only have 1 copy of a gene, then if it's a recessive, the recessive is most likely to be expressed when there's not a match. And in males, that's part of the problem. Women would, and so there's some color blindness that works like this too. Females have 2 copies of a gene, two alleles, and men have only 1, and so if the recessive is passed along, it would be expressed in a male, but in a female, in a heterozygous female, it would not. There's another form that is autosomal, and so it's seen equally in both sexes. Thrombosis, so that's what we talked about with thrombin. Abnormal clotting here in unbroken blood vessels, and that leads to a clot. And in many cases, especially for people who are relatively inactive, this happens in veins. And so they aren't moving a lot of blood. And you know that veins are very much a low pressure system. And if you're standing up or if you're sitting up and there's not a lot of movement in your legs, then the blood tends to pool in your legs, and that contributes. Already we've talked about low blood flow to an accumulation of thrombin and potentially a thrombus, which is a blood clot. So you can see about 1/4 of people over age 50 experience this. It's one of the many, many reasons why my wife and I are choosing to take a lot of walks. So we walk 2.3 miles. I've probably told you this 100 times. I'm going to tell you more because I'm really proud of it. 2.3 miles, and we did 235 of those walks for about 500, and I think, 530, 540 miles last year. And we've already done 25 walks this year by February 16th. So we've been doing, it's 18th today, but that was the last walk. So we're doing a lot of them even through the winter. What that does is moves an awful lot of blood through our legs. And it helps our heart and circulatory system and cardiovascular, pulmonary, just as well systems to stay healthy. But, for various reasons, people have injuries or otherwise they choose not to be very active, and that has health consequences, guys. So what is an embolism? An embolus is anything that is traveling in the circ system that has the potential to plug up blood vessels. And you can imagine, just like flushing something big down a toilet, like a tennis ball, although it initially might go down some of the pipes, when you find a narrowing, it's got the potential to clog things up. Okay? And so where we have blood distributions from big blood vessels to littler blood vessels, which is the blood flow from the heart going out to, well, actually the heart itself, or to the brain, or to the lungs, or the muscles, those blood vessels, those arteries, become smaller into arterioles and then eventually capillaries. So if you've got an embolus, a great big clotty thing that's circulating fine through the big blood vessels, by the time it gets to a smaller blood vessel through the branching system, it can clog things up. And that, of course, could lead to a cutoff of blood supply, and it could lead to the death of those tissues that are affected by that clogged blood vessel. This is often the cause of strokes and heart attacks. And so what we worry about are narrowed blood vessels and those narrowing areas. So it might just be a narrow passage, like having a bunch of stuff in a hallway where everybody's got to traffic through a more narrow area. Those are areas that are more likely, we think, to clog up by an embolus. So, more likely to occur in veins than arteries because blood flow is slow. Just talked about that. Blood flow is slow going back to the heart, and so you could get an embolus developing in the veins. The veins, now this is important to know, more likely to be where they form, and as blood flows back to the heart, it goes from smaller to larger. So those clots are unlikely to be clotting up veins. Once it gets back to the heart, it goes off to the lungs, and you know that in the lungs we have smaller and smaller blood vessels, and so what you can have is a blood clot in the lungs, and that can be life-threatening. It's called a pulmonary embolism, blocks circulation to the lungs, and you can die of it. This is me making noise with my soda can. So what happens if we cut off the blood supply to the heart? Well, it's called a myocardial infarction. And an infarction is the death of the tissues. That also can happen in a stroke. So if we cut off blood supply to a specific part of the brain, that part of the brain can die. And sadly, at least at this point in modern medicine, we cannot repair that area of the heart that's died or that part of the brain that's died. So what has to happen is, in recovery, you have to have other tissues compensating. So in the case of a stroke, so part of the brain was lost, you're going to relearn things. You're going to use another part of your brain to take over. So the recovery from a stroke can take a long time as a person learns something new. As far as the heart goes, in a myocardial infarction, it's just the other heart muscles have to work harder. And that means the cardiac output, we're going to talk about these things here in the couple weeks to come. Cardiac output is simply how much blood is the heart pumping. Well, if the heart has lost some of the muscle, its capacity will be reduced. So this kind of situation could lead to a lot of deaths, and does, and cardiovascular disease is a very serious threat to people in the United States. So what might we do to worry and to manage a little bit of blood clotting? Well, vitamin K, if you had looked at all those different factors that are in play here on the coagulation pathways. Sorry, I was just looking for the page number for you, but that was on 697. I'm just looking to see if vitamin K was listed there as one of the factors. I'm not finding it right now of where it was listed. In any case, vitamin K required, and so drugs like coumarin and warfarin, which is called Coumadin, these are vitamin K antagonists. So if vitamin K is needed for clotting, then if you interfere with K, you can reduce clotting. This is what I was telling you about, all those details of the clotting pathways. I mentioned aspirin, and here now specifically, you could look in thromboxane A. I'm just looking to see if thromboxane A is listed here. I'm not even seeing that in that detail. Yeah, so this is even beyond the textbook figure. But vitamin A suppresses then something that's important in this cascade of reactions. And we've discovered all kinds of other anticoagulants. I've mentioned to you, if you had zoology, that leeches, medicinal leeches, have an anticoagulant called Hyridin. And you may not know that one of the properties of snake venom that makes them so dangerous is that they have an anticoagulant. And so what this will do is promote bleeding. And that, of course, is a real serious problem. Here, figure 18.8. And 18.8 is on page 700. Nice even 700. On page 700, here is a list of some of the terminology that you need to know and understand as the end of chapter information. That's important for you. Yes, some of the terms that are important for you to look up. And here is a list of some of the other kinds of problems that people can have on disorders of clotting. Or our other factors is here. It's not just clotting. So here they're talking about bacteremia and other kinds of things. And this is the end of our look at blood. So next, what we're going to talk about is the heart. And I had hoped that we would have gotten to the heart. Well, today's February 18th, Friday, February 18th. I wanted to get to it on Monday, the 14th of February. So I guess you have to guess, why did I want to talk about the heart on February 14th? And because it's Valentine's Day. But we missed it, darn it. Nonetheless, we'll talk about it in class here in the upcoming week. So this might just be the last recorded lecture.